Now we move into the third part. We have seen that who are they and where do they live. The question comes that how do they live, right? So they live because, as I said, that can have a very distinct metabolic diversity, and they can have a very distinct ecophysiology, right? Many of us are aware that the microbes can be can be utilizing light and carbon dioxide and perform the photosynthesis, right? So that we can classify in phototrophy, right? Phototrophs, which can do the phototrophy utilizing the light and inorganic carbon source, that is carbon dioxide, for the photosynthesis, right? So one example is rhodobacter. The second is that instead of using light as a source of energy, can use chemical as a source of energy, right? But fixing inorganic chemicals, right? Maybe even the carbon dioxide, right? That could be chemolithotrophs. And then there are microbes which not only uses the chemicals for the energy, but also uses organic carbon, right, for the metabolism. Those are chemoorganotrophs. Okay, I hope the definitions are clear. One which is using light is phototrophs. One which is using chemical are chemotrophs. In chemotrophs, you have two classification. One using the organic chemicals is chemoorganotrophs, and one using the inorganic chemicals are chemolithotrophs, right? In the phototrophs again, there are microbes which needs or which can do an oxygenic photosynthesis, or which can do oxygenic photosynthesis. It means that in due course of photosynthesis, it is possible that oxygen is produced, right? Which is mostly in the case of, let's say, microalgae in the photic zone, and then also cyanobacteria, that Prochlorococcus and Cinococcus, and then utilizing light, okay, doing phototrophy, but does not produce the oxygen in case of anoxygenic photosynthesis, right? So in this particular anoxygenic photosynthesis, it will not produce the energy, okay? There are several lineages. I will just give you an idea that purple sulfur bacteria or green sulfur bacteria, those are the bacteria which does the anoxygenic photosynthesis. We will see in more detail when we will learn about the marine bacteria, maybe tomorrow. Okay. So for now, what you need to understand that there are metabolic diversity and there are ecophysiological adaptations in the microorganisms in order to sustain in different part of the ocean. They can utilize light source, they can utilize light as a source of energy or chemical as a source of energy, utilizing the inorganic carbon or utilizing the organic carbon, right? If they are using organic, then these are chemoorganotrophs. If they are using inorganic, then they are chemolithotrophs. If they are using if they are producing oxygen, those are oxygenic photosynthetic organism. If they are not producing oxygen, those are anoxygenic photosynthetic organisms, right? If the organism, so mostly in most of the cases, that could be, or there could be exceptions as well, but in most of the cases, chemolithotrophs and phototrophs are autotrophs, okay? Whereas chemoorganotrophs are heterotroph. So in heterotroph, the cell carbon is obtained from one or other organic compounds, right? Whereas in autotroph, it uses carbon dioxide as a carbon source. So this is a major difference between autotroph and heterotroph, because in heterotroph you use mostly carbon dioxide as a carbon source. In heterotroph you use organic carbons as a form of for the carbon sources, right? And you can utilize light as energy or you can utilize chemical as energy, right? So I hope that this clear that there are different varieties of the bacteria, of the microbes present in the ocean, and they are occupying the different area because they are specialized in the different metabolic diversities, right? It means they have the metabolic diversity. If it is on the surface, mostly they will be utilizing the sunlight, and then they will be releasing the oxygen, right? Which we have seen that has been very important in the formation of the life on the earth, because this oxygen only brought oxygen into the atmosphere, right? Now, the chemolithotrophic activity, we can also see in a way that whatever the nutrient cycle we are seeing, be it a sulfur, be it nitrogen, be it carbon, be it whatever, iron, whatever cycle we are seeing, because of the chemolithotropic activity of the microbes, right? There are diverse mechanisms, and that's why they are able to do the transformations of carbon, sulfur, and nitrogen, and that is actually what is the microbial nutrient cycling, right? So basically, you utilize inorganic energy and electron source, and then produce the oxygen through the electron transport chain, right, and produce the ATP, right? So this is the chemolithotrophs, okay? And then in case if you are using same inorganic carbon source, but then using light, then that is photolithotrophs, or you can also say phototrophs, right? So in a nutshell, what we can see, we can divide into photoautotrophs, we can divide into photoheterotroph, chemoautotroph, and chemoheterotroph, right? So here photolithotrophic. So photolithotrophic is mostly when you are using the light as a energy source and inorganic carbon as a carbon dioxide as a carbon source. Photoorganotrophic when you are using the light as an energy source, but organic compound as a electron or the hydrogen source, and organic compound as a carbon source. The examples are purple and green sulfur bacteria. Similarly, if it is chemoorganotrophs, so chemoorganotrophs, you are using organic compounds for both as energy and a carbon source. And in chemolithotroph, you use carbon dioxide and the chemicals for the energy source as well as the carbon source, right? So this is the overall varieties of the microbial diversity in the marine environment, because they have adapted, or they have the capability to utilize different energy sources and utilize the different organic and the inorganic chemicals. They are adapted in the different parts of the ocean, okay? Uh, so do we only need the carbon dioxide and the sunlight, or do we only need the uh, chemicals for the energy and chemicals for the carbon, or we require anything else for the survival and growth? So you may have heard, or you may be knowing very well, that for any life forms to sustain, we also require a lot of macronutrients and micronutrients for the growth and the survival, right?
So as I was saying, that not only the light and the CO2 is required for most of the microbes, right? There are other inorganic nutrients are also required. If you can see that many of the species, in particular to diatom and dinoflagellates, are auxotrophic in nature. So what does auxotrophic means? Auxotrophic means that the organism may lack the biosynthetic pathway needed for specific compounds, and hence they need to obtain from other organism in the food chain or through the acquisition, right? So there are, as I said, that there are microbes which may not be able to synthesize, or which may not be able to, use, which may require the nutrients apart from light, CO2, and few elements, right? For example, half of the surveyed species of algae requires vitamin B12, that is cyanocobalamin, for their growth and survival, right? The availability of vitamin depends on overall microbial community composition, or there are tight association between the microalgae and the bacteria, such as Rosiobacteria, Merinobacteria, Sulfobacteria, and their relatives. This has major effect on the regional and the seasonal diverse distribution and the activity of phytoplankton. So how do the microbes gets these nutrients from the environment? As you can see on the screen, there is a possibility of direct diffusion into the cell, wherein the nutrient in the surface water can be uptaken directly by the cell. Diffusive process alone can fuel the growth and the productivity of the abundant free-living unicellular microorganism in the open ocean. However, we also know that not only the direct diffusion, but there are facilitated diffusion, which you can also see as a passive transport of the nutrient from the environment or from the surrounding water happens into the cell. So these transport or these nutrient acquisition for the microbial growth can happen via the transport proteins in the cytoplasmic membrane. So active transport of the nutrient require a dedicated solute transport system and input of the cell energy. There can be several different type of the mechanism, especially for the molecule uptakes and type of the proteins present. There are several examples in this case. For example, ABC type transporters, there are symporters, there are antiporters, there are group translocation transporters. You can also see on the screen, you have various different examples of or various different transporters shown in the cell in the cytoplasmic membrane, right? So further details can be seen as, for example, in the Sphingomonas species A1, ATP binding cassettes transporter, that is ABC transporter, ALG M1 M2 SS, is responsible for the import of alginate across the cytoplasmic membrane. So now we know that the nutrient acquisition from the surrounding waters in the microbial system can happen either through the direct diffusion or through the passive transport or indirect diffusion, right, or the facilitated diffusion, and that can happen via the transport protein into the cytoplasmic membrane. There are various different examples of the transport systems on the cytoplasmic membrane. For example, ABC type transporters, importers, antiporters, group translocation transporters, and so on. Further, if we look, let's say one particular example about the SAR11, which is which I've also mentioned earlier, that it is one of the group of a small carbon oxidizing cells that reach a global estimated population size of 2.4 into 10 to the power 28 cells. That is approximately 25% of all the plankton, and they are found throughout the ocean, but reach their largest number in stratified oligotrophic gyres, which are an expanding habitat in the warming ocean, right? So if you can look into the SAR11 bacteria, there are or the genome of the SAR11 bacteria, you will see that there are the genome imports individual transporters that import various organic compounds that includes amino acid, DMSP, taurine, polyamine, simple sugar, and phosphonates. And inorganic compound, for example, iron and complex phosphates in the genome of SAR11, right? Further, proteins of another system, that is tripartite ATP independent transporter trap, which use an electrochemical gradient to drive the nutrient uptake, are also very, very common. In the table here, you can see that the genome of the SAR11 are having products which is being encoded by various genes, and they have various roles in the nutrient, organic and inorganic nutrient uptake from the environment. Further, iron acquisition which is considered as very important for the microbes. It is being facilitated by the siderophores. The question comes like why the siderophores are evolved, because of the lack of the free iron in the open ocean, and it's almost undetectable. So in the marine water, iron occurs in the ferric state bound tightly to the organic ligands. So it is difficult for the marine microbes to obtain enough for their growth. Hence, marine microbes releases chelating agent, which is also known as siderophores, which binds to the iron, prevents its oxidation, and allow active transport into the cell via specific receptors which are present on their cell surface. Right now, over 500 chemicals are identified as siderophore. Some of the most common, if you see the example in Vibrio vulnificus or Vibrio angularium, which produces siderophores, are known as vulnibactin or ogibactin, and these are very important factor even in the virulence, right? So what do we learn? We learn that the iron in the marine microbes is or facilitated by the siderophores, and they are basically a group of, or they are basically the chelating agent that binds to the iron, prevents its oxidation, and it allows the transport into the cell via the specific receptors on the cell surface. Further, there are a couple of more concepts. Concepts are there. One is copiotrophs and oligotrophs. So what is copiotrophs? Copiotrophs are those organisms which are found in environment rich in nutrient. For example, if we look into the estuaries, where you have very high level of nutrient conditions, right? So microbes grows very rapidly when exposed to these kind of very high nutrients or very high substrate. And example in the vicinity of the phytoplankton, for example, Photobacterium angustum is 14, right? Whereas oligotrophic are those oligotrophs are those organisms which are found in the oligotrophic water. Now, with the oligotrophic, what does that mean? It means that it's a nutrient poor environment. If we look, most of the open ocean, deep oceanic sediments, caves, glaciers, polar ice, deep subsurface soil, aquifers, ocean waters, and leach soils, these all are the example of the oligotrophic conditions. It means that the nutrient poor environment, and there are many organisms which are adapted to live in such nutrient deprived environment. Many bacteria have evolved to grow optimally at either high, that is copiotrophic condition, or low, that is oligotrophic nutrient concentration. And that is also one of the factor that enables different species to colonize distinct trophic habitat in the ocean. You can see one of the colorful image on the screen, which was published in PNAS, that marine bacteria trophic strategies, and you can be able to see the examples of yellow extreme oligotrophs, red moderate oligotrophs, green and blue moderate copiotrophs, and cyan color as extreme copiotrophs, right? So again, examples you can see on the screen, different bacteria or different microbes which are oligotrophs or the copiotrophs. One of the factor or one of the thing about the copiotroph is that they show feast or famine life like lifestyle. What does this feast or famine lifestyle means? It means that rapid growth rates and increased cell size can be observed in response to the increased level of the nutrients. They often produce free exoenzymes and highly diverse nutrient transporters, and have relatively large cells that often show mortality and chemotaxis in response to nutrient gradients. Whenever the condition or whenever the nutrient decreases, when the condition is not very favorable, these copiotrophic microbes reduces its size during the cell division, and various other adaptation it shows in response to starvation. So they reduce the synthesis of the macromolecules. Size reduction happens during the cell division and other adaptation to the starvation condition. There are several studies published where it is shown that the copiotroph shows the feast and famine lifestyle. One such study you can see on the screen, where experimental evidence for the long-term coexistence of copiotroph and oligotroph bacteria in the pelagic surface seawater was studied. How would you find if the strain you culture is copiotroph or oligotroph? That's a very interesting question. And if you really want to find out if your culture is a copiotroph or oligotroph, there are few things which you have to look. For example, copiotroph will have relatively larger genome size, higher proportion of the extra cytosolic proteins, extracellular proteases, peptidase, chitinase, amylases. Also, the copiotroph grows very fast if you are providing the high nutrient rich condition, while in the less nutrient condition, it will be so slower. Whereas oligotrophic comparatively will have smaller genome size, almost no or less extracellular enzymes, and have constant low rate of growth. So in future, if you are having a marine microbe and if you are growing them, and if it is slow in the growth, you can say that these bacteria or these microbes could be oligotroph type, whereas those which can grow very fast if you give high nutrient and slow if you give low nutrient, that could be the example for the copiotroph. Another very important concept is a viable but non-culturable state, VBNC. In the environment, many a times it happens that when you are working with the marine microbes or in general with the microbes, you will see that the cell may not be able to culture, right? So one of the factor in those cases is viable but non-culturable state of the microbes or the bacteria. Sleeping cell, which was introduced in 1980 by Rita Colwell and the co-workers, who observed that the Vibrio cholera does not die off when introduced into the environment. And later, it was shown to occur in a wide range of gram-negative and a few species of the gram-positive bacteria, including the pathogens of the human, fishes, and the invertebrate. Now, these VBNC cells have been defined as a cell which, induced by some stress, become unculturable on media that would normally support their growth, but which can be demonstrated by various methods to be alive and capable of returning to metabolic, active, and culturable state. Okay, so so you can see even in the graph that the cells or the bacteria undergoes VBNC at the time of the stress, like antibiotic, and as the antibiotic is removed, the cell has returned to its viable state, right? So as the removal of the stress happens, there is a possibility to get the microbe or the bacteria cultured back into the plates. Why the VBNC happens? It's not very well understood, but could be possibly, as you can see on the screen, where there is a possibility that the activation of the REL-A and SPOTB due to amino acid starvation leads to the intracellular accumulation of the alarmone, that is PPGPP, right? That may lead to the inhibition of the exopolyphosphatase, causing their accumulation due to continued activity of the phosphate kinase, polyphosphate kinase, and alone protease, which is activated further by polyphosphatase, and then that begins to selectively degrades protein, including the antitoxins. Now, this disturbs toxin-antitoxin ratio and allows free toxin to inhibit the cell growth, leading to persistence. And it is important to note that even if that exact mechanism is not at play, each component has independently been shown to have a significant role in the VVNC condition, or viable but not active condition in the microbes, right? So even though it is not fully understood, but possibly this kind of mechanism may happen where the free toxins is not being bound with the antitoxin within the cell, and this free toxin can cause the inhibition of the translation and the cell growth, and that can induce the persistence as well as the VVNC state in the microbes. So that is very much possible.
Only in a regular or mesophilic environment, microbes in the marine environment are also present in various different extreme conditions. There are examples of various extremophilic environment, for example, high saline conditions, salt pan, hypersaline lakes, hydrothermal vents, deep sea cold environment, high pressure deep sea environment, colder polar environment, and so on. So microbes do have the capability or do have the adaptation through which they can survive in those hostile environment where it is very difficult for higher organisms to survive as such. I'll give you some of the example of those extreme microbes and how do they survive in those extreme conditions. For example, the very first one is life in a high salt concentration with changing environmental conditions. This is one of the example from our own lab where we studied moderate halophilic bacteria, that is Selinovibrio species, and we can see that the bacteria have developed the different mechanism to survive the high salinity conditions. They produces compatible osmolytes which provide them the osmotic balance without interfering with the metabolic functions of the cell. They produce deuteric ion solutes such as glycine, betaine, ectine, uncharged solutes such as trihalose, and anionic solutes such as glutamate and hydroxyl butyrate. All these act as an osmoprotectant for the bacteria. They also have developed the cell envelope modifications where increased negatively charged phospholipids are abundantly present on the cell envelope, helping them to uh, sense the variable salt concentration in the growth media, and also they have the internal ion homeostasis mechanism where a lot of sodium potassium antiporters, NADH oxidase, chloride transporter, potassium transporters are available, and which helps them in maintaining the internal ion homeostasis.
Similarly, since the bacteria mostly in the photic environment or mostly in the intertidal area exposed on the sediments during the intertidal time, or also in the salt pans or hypersaline lakes at the time of the low tide, or when there are less waters, they do have the a number of UV repair, I mean DNA repair mechanism, which could be because of the damage of, I mean, which could help them to repair the DNA because of the radiation stress. Okay, and you can see a number of, so close to 15 or 16 various pathways or genes involved in these pathways were identified in the bacteria if you expose them to the ultraviolet radiations, mostly UVA and UVB, right? So it again suggests that the bacteria do have uh, even though many of them have a very reduced genome, they do have potential to encode the genes involved in the metabolic pathways to preserve them or to protect them from the ultraviolet radiations, which is involved in the DNA repair. Also, one of a very striking feature of the bacteria in the marine environment is the bioluminescence, right? We know that a number of bacteria does have the operons which help them to bioluminescence. So this bioluminescence is also a factor which could be derived because of the stimulation of the photoreactivation. We know that photoreactivation is a very common mechanism to repair the DNA in response to ultraviolet radiation or in response to any DNA damage, right? And now that this photoreactivation requires light, if the bacteria are, I mean, if the bacteria at the time of night or if there are not sufficient light, they have developed or there is a evolutionary drive for bacteria to carry on the bioluminescence, which could be helping them to repair the DNA in case of DNA damage. Okay, so these kind of mechanisms are also available in the microbes in the marine environment. Now, also, uh, as we already have seen, that most of the marine environment below 200 meter is very cold, below the thermocline layer, right? And also a majority of the oceans, close to 70% or 80% of the water is existing or is present in the cold conditions, right? So most of the bacteria, we can say that, are cold adapted because the water column below 200 meter is huge and microbes are present everywhere. So they also have several adaptations in order to live in the cold conditions of the ocean, right? For example, the oxygen solubility, if it increases in the cold in the deep sea, right, that can lead to the oxidative damage because of the production of reactive oxygen species, right? So all the microbes, if they want to survive the oxidative damage or oxidative stress because of the reactive oxygen species, they do not want to continue with a mechanism or oxidative metabolism, which again produces the oxidative metabolism, right? For example, glycolysis or pentose phosphate pathway, or also TCA, electron transport chain, right? So these microbes depresses this oxidative metabolism and instead have adapted to secondary metabolic pathway for example, ethanol oxidation or glycosylate pathway or methyl glyoxal, right? Uh, in addition to that, these microbes also have a number of cryoprotectants which help them to adapt in the cold condition. For example, glycine, betaine, glycerol, trehalose, sucrose, mannitol, and sorbitol, right? So what happens is that these cryoprotectants, they decreases the freezing point of the cytoplasm, and then it prevent the macromolecular aggregation or scavenge the reactive oxygen species in the cell, right? So these kind of adaptations the marine microbes have acquired in order to survive in the low temperature conditions in the deep ocean, right? Similarly, if we see into the deep sea, there is a huge pressure. If you can see my screen, that what you see is at the 0 meter or at the surface, you have 1 bar of atmospheric pressure, right? As you go every 10 meter, there is an increase in 1 bar of the pressure on any body, right? As a result of which the volume of the air in our body also reduces drastically, right? So this you also have to keep in mind when you do the scuba diving, because after every 10 meters there is increase in the pressure, right? And that affects the volume of the air in our body. So that is why we say that if you suddenly come up to the surface from 30 meter, let's say, then there is a chance that all the air cavities in the body will go burst, right? Similarly, if the microbes are staying in the in the deep sea, they are subjected to the pressure stress. That is the barophilic condition, right? that is also called as piezophiles, because those microbes which can tolerate the pressure. And also you have the sacrophilic condition. It means that the temperature is low and the pressure is high, right? Though not much mechanism has been studied or understood, most of our understanding is coming from these two particular bacterium, that is a photobacterium profundum and the 7L species, right? And what we know about these two, that they encode some certain proteins on the cytoplasmic membrane, right, which is similar to what we have in the Vibrio cholera uh, uh, proteins, which helps in the detecting environmental conditions. So whenever a cell undergoes a stress, or which whenever a cell senses an environment change, right, there should be some sort of mechanism to sense that changes. So these bacteria do have cytoplasmic membrane proteins which regulates depending on the after sensing the changes in the environmental conditions. So that could be linked with the changes in the pressure or temperature or maybe salinity or whatsoever the conditions are, right? Similarly, in the deep sea bacteria, there are also phospholipids and lipopolysaccharides which are modified and which help them to maintain the membrane fluidity. Okay, because also the pressure is huge, so the membrane fluidity should be maintained in order to perform the cellular functions in the deep sea. Right. Most of the bacteria, at least in the deep sea, belongs to gram negative, because they also have outer membrane and the lipopolysaccharides, right? So extra cushion in the cell that helps them to survive the stress, and also helps them to attach to the surface of minerals or other organisms in the deep sea. Okay, and again, a lot of variety of transporters are there in the deep sea bacteria or deep sea microbes, which helps in the nutrient acquisitions, right, respiration sensing, signaling. It is very much specific. Okay, most of the bacteria in the deep sea again are very less motile, possibly due to the perturbation of the cell membrane and the flagellum apparatus. Some of the bacteria do also produce a second type of flagella, that is a lateral flagella, in response to high pressure or low temperature conditions. Okay, so we have seen that the microbes do have the adaptation strategies to survive in the high salinity area, low pH, low temperature area. Okay, and also to the ultraviolet radiations. There are also adaptation in the hydrothermal vents, right? So now in hydrothermal vents, what are the conditions? So the first condition is the high pressure, right? And second is that there could be in the outer part of the vents you have the low temperature conditions, and in the near part of the vents you have the high temperature conditions. So all these stress conditions are working at the same time, which distributes or which defines the microbial community present in the deep sea hydrothermal vents. Okay, in order to tolerate with the salinity stress, because near to the vents you may have the uh, sediments where you can have a varying salinity, and because of the intrusion of the gases from the vents, you can have the salinity variability. And in order to maintain that, you do have, microbes do have several transporters like potassium, sodium, potassium chloride transporters to maintain the salinity. Also, you have a lot of osmolytes similar like sugar, amino acids, polyols. These are the similar compatible osmolytes which help the cellular membrane to remain in a fluid form and does not affect the osmotic does not let the cell go osmotic stress. Okay, also, there are two specific compatible osmolytes, that is manosyl glycerate and then this dimyoinositol phosphate, right? These are exclusively accumulated by the thermophile bacteria in the hydrothermal vents. Okay. Some of the pressure tolerant bacteria also accumulates the hydroxybutyrate in response to the pressure. Okay. This particular vents do have the bacteria and the archaea, which changes the cell membrane from the gel phase to the liquid crystalline phase and then to the fluid phase, depending on the different temperature or the pH they senses in the environment, right? So as I said that these bacteria are or these microbes do have a sensing mechanism because of the receptors on the cytoplasmic membrane, and as soon as they sense the changes in the environment, they undergo the membrane fluidity changes, right, from the gel phases to the liquid or to the fluid phases, and all are like interconvertible depending on what kind of atmosphere, or I mean what kind of environment they are facing, whether there is an increase in temperature or pH, or temperature or pressure, or temperature or pH and decrease in pressure or decrease in temperature, right? So all these three forms can interchange with each other, right? Now, as I said that depending on the kind of the winds and depending on the locations of the gases coming out from the winds, the microbial community changes, right? So even though the hydrothermal winds are present worldwide, the microbial community structure are shaped very much locally. For example, you can see in the rising plume, you have a different kind of setup, and whereas you have in the neutral buoyant plume, where the plume is going in this direction, not in the straight direction, you can have a completely different setup. Mostly in this case, where the neutrally buoyant plume is going, right? So in the plume you have a variety of microorganism which have the potential to utilize the manganese and iron, right? And this manganese and iron can go as a biotic metal oxidation into the sediment, causing this metalliferous sediment, and that can lead to microbial mats as well, right? At the same time, you have the bacteria, which is either free living or attached symbiotically with the riftia, right? And then performing the maybe here you can see performing the utilizing the sulfur and making the either the carbon which is going into the open sea, right? Or otherwise they are also releasing the siderophores ions, which is again further going into the open sea. So likewise there is a ecological and biogeochemical interactions at the deep sea hydrothermal vents, depending on what kind of vents are there, whether it's a rising plume or whether it's a neutrally buoyant plume. Okay. And again, if you can see that within the same hydrothermal vent, wherever the temperature is higher, you have a specific variety of microbes occupied in that particular area, utilizing the different set of energy, electron donor, and then a different set of electron acceptor. Okay. So mostly methanogenesis are happening where in the hyperthermophiles and the thermophile region, whereas in the mesophilic condition, where of course in the hydrothermal vents also you can have the gradient, right, from where the vent is starting or going into the open sea. Now if you go a little further, it's a kind of a mesophilic environment, and then a different set of bacteria is been utilizing different chemicals for as an electron acceptor and electron donor. Mostly it's an anaerobic environment. Further you go up, and then the temperature reaches which is equivalent to what is there in the deep sea, that is below 4 degree, right? And that is a place where psychrophiles are dominant and mostly aerobic, and utilizing sulfur as an electron donor and oxygen as an electron acceptor. Okay. So different set of bacteria will be are adapted in the hydrothermal vents. Okay.